Hey guys, this is a very fundamental kind of initial discussion of the immune response and the different ways that the immune system can respond. We're going to go way deeper than this, but it's important that you understand at least this level at first. So basically here is a cell, and you can see here that these little things around here are pathogens. And so you know, these pathogens could be viruses or whatever, doesn't really matter what they are, but they get into the cell, and clearly from this point the cell to some degree is able to destroy parts of these. I'm sure the rest of them are probably wreaking havoc on the cell, but what the cell is going to do is this. It's going to take a little teeny piece of the virus or protein from the virus or bacteria, if that's what it was, and it's going to display it. And there's two different display mechanisms. One for self-antigens, and again, antigen is any substance that causes an immune response. And so clearly, since an immune response is happening, that's why this little part of this bug or virus is called an antigen. In some people it could be peanuts, or in my case it could be penicillin. It doesn't have to be a germ, it's whatever your body thinks is foreign matter. So in this case, it's going to create what's called an MHC, major histocompatibility complex. And this is an MHC class 2, and that is basically non-self antigens, which means that this little piece here is clearly not part of this guy. It's almost like this cell is saying, help! And so because the cell is displaying this, these antigens, it gives off a chemical that brings in other cells. And basically the helper T cell is one of the major ones that responds to this. And so you can see here that the helper T cell is binding to the MHC class 2 of this particular cell that's in trouble. And from this point what happens is the cell that's being invaded gives off a chemical called interleukin 1, and then the T cell gives off interleukin 2. And interleukin 2 is more of like a call to arms. And it's going to be calling over other white blood cells. So from this point, a couple different things can happen. There's basically two different pathways of what can happen. One is called a cytotoxic T cell, and this particular mechanism does not involve antibodies. You probably read a fair amount about that in chapter 12. So this is one particular way that the body can destroy that virus or the cell that has the virus in it. That does not involve antibodies. And the other one involves B cells and the production of B cells. This one is the mechanism by which our bodies and dogs and cats and pretty much everything else that produces antibodies, how they make them. So we're going to take a look at kind of both of these really quickly. The first one would be the cytotoxic T cell and how that kind of thing happens. So here's a normal cell, right? And again, it gets attacked by a virus or a bacteria, and it displays the antigen in an MHC class 2. And so because of that, now the cytotoxic T cell, smelling, for lack of a better word, interleukin 1 that the cell's given off, and the other cell damage will basically bind to this MHC class 2, like it's doing right now, and then it's going to give off chemicals that will literally destroy the host cell. Basically are called cytotoxic. That word should make sense. It's chemicals that basically destroy the host cell. But the good thing about destroying the host cell is that you've also destroyed the virus in it, or at least the bacteria in it, and at least not given it a mechanism to reproduce. So that's one whole system of how you can produce or rid your body of pathogens. However, this is the way more common one I'm going to show you now. This is a very fundamental view of how your body makes antibodies. So, and that's of course a B cell. So what happens now? If you recall that we had the helper T cell, and what they skipped in this video was that there was a cell that was damaged. It called over the helper T cell. The cell that was damaged gave off interleukin 1, which caused this particular helper T cell to give off interleukin 2, which basically then allowed this interaction between the helper T cell and the B cell. And so at this point, the B cell is going to become activated. As you can see here, it's going to start to divide a lot. This particular B cell was chosen because of something called clonal selection, in the sense that the particular antigen fit this type of precursor B cell, so it gets chosen to divide over and over and over again. And that's why with patients, one of the ways that you can tell if somebody has an infection is you can kind of palpate or feel under their neck. And if their lymph nodes are swollen, part of the reason that's the case is that the body is actually creating massive amounts of B cells and neutrophils, which are kind of like soldiers, to fight infections. So that's one of the ways that you would know. So once these B cells divide like this over and over again, they produce massive amounts of antibodies, and these antibodies are specific to antigens. So the reason you also need a flu shot every year is that the flu virus mutates enough that these little antibodies that are basically like chewing gum that stick to things, they stick to bad germs. The germs can change just enough year to year that these pieces of bubble gum aren't going to stick to last year's germs. And so that's the idea about why we tell people to have a flu shot every year. So we can see all these antibodies now floating in the lymph system, in the bloodstream, pretty much everywhere. And what they do is they stick exactly to this particular antigen. And what that does is twofold. A, it means that now the virus or the bacteria cannot bind to host cells and get in and cause trouble. And it also clearly identifies this thing as being a foe. It doesn't belong here. And so then other cells like a macrophage you're going to see in the corner can identify this particular bug as being foreign and destroy it. Now, after you've spent 4 or 5 days or 6 days having the flu, for example, and getting sick and having your body do this process of antibody production to destroy it, it would seem like a huge waste of time. What if the exact same virus came back a month from now? You don't want to be sick again. So your body has to try to remember kind of the war. It's almost like the grandfather talking about what his days were like in the war. Well, the grandfather in this example would be a certain kind of B cell called a memory B cell. And basically this is the type of B cell that does not become a plasma B cell. It just kind of floats around the system for sometimes 10, 20, 30 years or longer. And if that bug ever comes back, it can just instantly start to divide and not have to go through the process of figuring out which antibody is going to be produced to stick to this. It already knows. And so that's kind of just like a generic, very initial discussion of immune response, and we will get into way more detail about that later.